Hello students, this is Dr. Ranjini. Welcome to e-learning session in Microbial Technology. In this session, we learn the biotechnological importance of microorganisms. Microorganism is a living thing that is too small to be seen with the help of naked eye. We cannot see them by our naked eye. The scientist Anthony van Leeuwenhoek invented the microscope. And this invention helped us to view these unseen organisms. The use of scientific and engineering principles to the processing of materials produced by microbes to create beneficial products, beneficial proteins, or beneficial processes for the betterment of human life is called microbial technology. What is microbial technology? We are using scientific and engineering principles for processing of the microbial product or for the processes which will be exhibited by the microorganism for the betterment of human life. That is called microbial technology. Let's see why you have to use these microorganisms for the betterment of human life. Microbes are ubiquitous in their distribution. We all know microorganisms are present everywhere. They are ubiquitous in their distribution. Not only that, they can grow on cheaper raw materials. We can grow these microorganisms on a very cheap raw materials. As we all know, the microorganism can grow even on dead and decaying organisms. So the food requirement is very cheap. The microbes generation time is very less. Under ideal growth conditions, bacteria can take 5 to 30 minutes. The growth of the microorganism, though it is highly complex and coordinated process, ultimately expressed by increase in cell number or cell mass. That is the growth. The process of the growth depends on availability of the nutrient or essential nutrient or requisite nutrient and their transport into the cells and their transport into the cells. Not only this, nutrient play an important role, but also many environmental factors also decides the growth of the microorganisms, like aeration, oxygen supply, and then pH of the media, and even the temperature where we are culturing the microorganism. So this doubling time, this generation time or the doubling time refers to the time period required for doubling the weight of the biomass, doubling the weight of the biomass. While the generation time represents the period for doubling the cell numbers. Period for doubling the cell numbers. Doubling times normally increases with increasing cell size and complexity. For example, as we saw here, the bacteria, they take around 5 to 30 minutes for converting one cell into two cell. And even in certain bacteria, this doubling or the generation time may go up to 1 hour. For example, yeast, they take 1 to 2 hours. Animal cells, they require 25 to 48 hours. Plant cells require 20 to 70 hours. So in general, when all the conditions are kept optimum, ideal for the growth of the microorganism, the supply of the nutrient will decide the growth of the microorganism. The next point is the microbial growth is season independent and can be easily grown and manipulated. Microorganisms can be grown in the laboratory condition under in vitro condition without any season effect. And next point is these microorganisms, few microorganisms, they depend on the other microorganisms for their food or for their life. But certain microorganisms have the capacity of synthesizing their own food material. So such autotrophic microbes have high efficiency of solar light conversion. That means they convert the solar energy into chemical energy in the presence of certain pigments, which play an important role in photosynthesis or conversion of solar energy to chemical energy. So that will be considered, or that will be utilized for the synthesis of the food material. Bacterial cells have extra chromosomal DNA called plasmids. In the cytoplasm, the bacterial cell has the extra chromosomal DNA called plasmids, and these plasmids can be manipulated to express the desired gene or to express the desired protein by recombinant DNA technology. Useful metabolites produced by the microbes are extracellular in secretion. This makes isolation process very easy. The important, useful, desired metabolites will be produced by the microorganism, and these products or the metabolites will be produced extracellularly. They'll be secreted out of the cell. And there are certain the metabolites which are intracellular in secretion, but the extracellularly secreted metabolite production will help in isolation process will be easy to isolate and purify such microbial metabolites which are extracellular in their secretion. Microorganisms like spirulina, chlorella, and other blue-green algae are rich in proteins and vitamins, could be consumed in dried forms. So these microorganisms are rich in proteins and vitamins. In addition to that, there are many useful the nutrients will be available in these blue-green algae, and they can be consumed in the dried form. So these microorganisms will be grown and their culture will be sun dried, and after that it will be consumed in the form of powders or in tablets, or certain food products will be prepared and will be consumed. Microbes are widely used in large-scale industrial processes because of their importance, nutritional importance. These are cultured in large scale in industry. They are crucial for the production of variety of metabolites such as ethanol, butanol, lactic acid, and riboflavin, as well as the transformation of the chemicals that help to reduce the environmental pollution. So these microorganisms, because of their capability to produce important metabolite, these are cultured in large scale and they are produced in industrial processes. Microbes can be used to create biofertilizers or to reduce metal pollutants. These microbes are proteinaceous in nature, rich in nitrogen components, and apart from that, they are also rich in many useful nutrients that will be utilized as the biofertilizer and also to reduce the metal pollutants. These microbes can also be used to produce certain non-microbial products such as insulin that will be used to cure the diabetes. So initially the insulin was isolated from the 4-year-old calf. Later it was produced in the pig to overcome the immunological reaction that will be elicited by this the pig insulin and other aspects. This insulin will be produced by the E. coli. Insulin will be produced by the E. coli. Nowadays, we are treating most of the diabetic patient using this recombinant insulin which is produced in the E. coli. Few microbes produces certain secondary metabolites like antibiotics, flavonoids, toxins are having high pharmaceutical importance in human life. Microbes not only used to produce the the metabolites which are required for the growth and development. Apart from that, there are certain microorganisms where they produce a certain metabolites in very low concentration to overcome the extreme environmental condition, or it may be the absence of the nutrient, or it may be the environmental condition. In order to sustain its viability, it will produce certain components or produces certain metabolites. They are called secondary metabolites, that includes antibiotics, flavonoids, toxins, etc. And these are having pharmaceutical importance. And hence these microorganisms will be exploited for the production of these secondary metabolites. This is regarding the biotechnological importance of microorganism, why we are using these microorganisms for the betterment of human life. So it may be for the production of certain proteins or desired product, or for the production of certain secondary metabolites, or it may be certain processes which will be beneficial for the betterment of life. That's why we are using these microorganisms. Next, we will move on to, so when we are using the microorganism in the bioindustry for the large scale production of these metabolites, which are having the importance in the human life, so we should see certain characteristic of the microorganism. When we are using in the bioindustry, the microbes use must be contamination free. That means when we are producing certain important primary metabolites or the secondary metabolites, it should be only particular microorganism should be growing. It should not be contaminated with other microorganism. For example, bacteria should not be contaminated with the phage. So like this, so when we are concentrating on the production of particular metabolite, only the specific microorganism should be there. And they should grow in simple media. They should not require the very costly nutritional requirement, should not be required by the microorganism. Minimal nutritional requirements should be useful in culturing the large scale microorganism further and their product. Also, preferably not required growth factors, that is, pre-formed vitamins, nucleotides, and acids. So, cheaper raw material, if they require for their growth, it will be easier to consider in bioindustry. The organism should be genetically and physiologically stable. So the microorganism should be stable. You should not change its genetic makeup soon after few experiment. Hence, they must resist random mutations. The organism, when we are using in bioindustry, should also accept certain degree of genetic manipulation to enable the creation of strains with more acceptable properties. When we are culturing the microorganism under laboratory condition, under in vitro condition, it should accept certain stable genetic manipulation for maximum production of the particular protein or certain pharmaceutically important metabolites. Microbes should grow vigorously and rapidly. The growth period must be shorter so that the repetition of the experiment or the production of the important metabolite will be easier. So this the growth period should not be more than 24 to 36 hours, because a certain metabolite production takes place only at the particular stage of the growth. So if the growth period is too long, it will be very difficult for us to isolate the product, or for the large scale production of the particular metabolite will be difficult. So these microorganisms should lead to a single desired product in a short time if possible. So it should not produce more than one product. So if it is contaminated, the separation or the purification will be very difficult. The product should not contain unwanted materials and other toxins. And microbes should produce their products extracellularly to reduce the cost price. So in order to make the isolation and purification make it easier, the microorganism should produce their product extracellularly. cellular. So it will be easy for isolation and purification. Microorganism should produce the product of interest in high yield. So it should produce the desired protein or the metabolite in high yield. Microbes used should be non-pathogenic. It should not cause any disease. The organism should not be too highly demanding of oxygen. When we are growing the microorganism, we usually grow them in the fermenter. You'll be learning about that in coming slide. So you should not require too much of oxygen. The microbes should be conserved for an extended period and become viable after thawing process. That means the microbes can be easily stored under lower temperature. So if you want to revive back, if you want to grow the same culture, you can just thaw the microorganism, you can bring them to the room temperature, and again you can grow for culturing them. So they should be conserved for an extended period and they can be stored and you can make them viable for the production of particular metabolite. This is regarding the few important characteristics should be served by the microorganism in order to uh, consider in the bioindustry for the large scale production of the particular metabolite. Next, we'll move on how these microorganisms can be cultured. We know microorganisms are ubiquitous in their distribution. You can isolate them from different sources like soil, water, or air, or even genetic manipulations are incorporated or recombinant by recombinant DNA technology in order to get the microorganism having the desired characteristic to produce the large scale primary or the secondary metabolite. So these microorganisms are cultured on the growth media is the balanced food supplement. It is supplemented with the sources of the carbon. So when you are growing the microorganism under the in vitro condition, you should give them the nutrient. You should have the balanced carbon source, nitrogen source, phosphorus, amino acids, trace elements, etc. And when you are growing the microorganism on the media or the nutritional supplement, the media should be sterilized. It should not have the contamination with the other microorganism. It has to be sterilized and then desired microorganism for the particular metabolite will be inoculated with the specific microorganism for the specific microbial product. So you can grow this microorganism on the solid culture or on the liquid culture. Solid culture, as we all know, the media will be supplemented with the solidifying agent like agar-agar or gel, right? So this will provide the adsorbing surface for the growth of the microorganism. So that is solid media. In liquid media, usually the broth, so it will be cultured in the form of the suspension and the media will not be supplemented with any agar or the gel, right? So it is just the liquid media without the solidifying agent and will be usually grown in the fermenters. So the fermenters will be used for the culturing the microorganism. Fermenter is a vessel designed to carry out fermentation process, that is biological reactions under controlled conditions. Hence it is also known as bioreactor. While designing the fermenter, several criteria which help in maximizing the yield are taken into consideration. That include, so the fermenters are designed for long-term operation in aseptic condition. Adequate aeration and agitation will be maintained inside the fermenter. Air will be supplemented or air will be plunged inside the fermenter. So inlet will be provided to this big furnace or the fermenter, and at the same time, the released air or the oxygen after it is utilized by the microorganism, after respiration, the air that is produced by the microorganism will be thrown out of the fermenter by the outlet. There will be pH control in order to maintain the ionic strength of the media inside the fermenter. And this fermenter will be provided with the central shaft to which the blades will be attached or the paddles will be attached. So that will be connected to the motor. So when we switch on the motor, this stirrer or the central shaft with the blade will move. So this will give the agitation or the mixing of the media takes place. So the homogeneous growth of the microorganism takes place inside the fermenter. Otherwise, what will happen? The cells settles at the bottom, they utilize the nutrient and they grow. But the media will be heterogeneous in production of the desired product. And the fermenter will be having the space or uh, the option for the transfer of the inoculum or the microorganism will be added, and this will be having the minimum labor in operation in harvesting or cleaning or the maintenance. Temperature control system will be attached to the fermenter in order to maintain the particular temperature. The minimum evaporation losses from the fermenter. It will be closed, and if the froth is formed, anti-foaming agent will be added in order to avoid the foam inside the fermenter. And this fermenter will be suitable for a variety of processes, and fermenter is provided with limited amount of medium containing all the nutrients at optimum environmental condition. And at the same time, in fermenter it will be provided with the outlet where you can collect the used media or the spent that will be collected frequently, and later it will be used for the isolation of the product or isolation of the cell, so that can be spent will be drained out in the outlet. So this is just the basic design of the fermenter, how it will be used to grow the microorganism under in vitro condition. When we culture the microorganism, the microorganism culture can be divided into open culture and closed culture. In general, it will be referred as batch culture. Then another one is continuous culture, and another type is it is just the modification of the batch culture, that is called fed batch culture. So microorganisms can be cultured in a closed fermenter, then it is called as batch culture. Here in this culture, microorganism will be inoculated to the known volume of the media. It will be inoculated into known volume of the media. Further, there is no supplementation of the media inside the fermenter, and the microorganism grow in the media supplemented in the fermenter until the nutrients are there in the fermenter. And once the nutrients are over or exhausted, or when the toxic metabolites are deposited inside the fermenter, the growth ceases. That means the absence of the nutrient, depletion of the nutrient control the growth. At the same time, the toxic metabolites secreted or deposited in the fermenter will inhibit the growth of the microorganism, as the fermenter is supplemented with the media, minimum volume of the media. Further, there is no supplementation of the media. From the beginning of the inoculation to the end, the microbial culture passes through several stages in case of the batch culture. If you plot the time on x-axis, if you plot the time on x-axis and the log number of living cells, that is the turbidity absorbance on the y-axis, you get the sigmoid typical curve like this. In case of batch culture, you have the media at time 0, you have the media at time 0, you have the media. To this you have inoculated few cells. As soon as you inoculate the cells into the media, the cell will not divide. Instead, it absorbs the nutrient and it will enlarge its cell size, and it will synthesize the protein and enzymes, nucleosides, nucleotides required for cell to undergo cell division. Due to that, you will get the same absorbance will be maintained for short duration. The length of this region depends on the type of the cell you have inoculated, number of the cell you have inoculated, and the stage from which you have isolated the cell. So this phase is called as the lag phase. It's actually the preparative phase for the cell to undergo cell division. And here the same absorbance will be maintained for few hours. And this is called lag phase. After lag phase, after the cell synthesizes the protein, enzymes, and other requisites for cell to undergo division, now the cell starts dividing. As the cell divides, absorption also increases. And here the nutrient, initially the nutrient concentration is very high, due to which the absorbance increases continuously. And the cell growth takes place in the number as well as in the cell size. And this is called growth phase or trophophase. It's otherwise called as log phase or exponential phase. It is exponential phase. After exponential phase, since there is no further supplementation of the media inside the culture flask or in the fermenter, the growth ceases. Cells will be viable, they will not divide. Cells will be viable, they will not divide. Due to which same number of cells will be maintained for very long duration. Since there is no supplementation of the nutrient inside the fermenter, same absorbance will be maintained. That is called stationary phase. That is called stationary phase. Further, since it is a closed culture system, there is no supplementation of the nutrient. There is no supplementation of the nutrient, due to which cell dies and the absorbance decreases. That is called decline phase. That is called decline phase or the death phase. So this typical sigmoid curve you will observe in case of the batch culture. It's a closed culture. There is supplementation of the nutrient only once. There is no further supplementation of the nutrient, and due to which the depletion of the nutrient will decide the growth of the microorganism. At the same time, toxic components or the metabolites deposited inside the fermenter will limit the growth of the microorganism. So this is the growth curve you observe in case of the batch culture, or it is a closed culture. Next, we'll move on to the continuous culture. Continuous culture is that where a steady exponential phase or the growth phase, trophophase, is achieved. Usually, growth retards due to depletion of the nutrients in case of the batch culture. But in case of the continuous culture, the growth will not be retarded. Instead, we will have the steady exponential phase, or the growth will be maintained. Rather than the accumulation of the toxic products will be removed by removed from the fermenter in the form of the spent that will be removed, due to which the toxic substances will be removed out of the fermenter. That also will promote the growth of the microorganism. As there is the intermittent supply of the nutrient inside the fermenter, it is called open culture. The continuous culture is called open culture. And here the supplementation of the nutrient as well as the removal of the spent, which is filled with the toxic substance, will maintain the cell in the steady exponential phase. So the microbial biomass will be continuously produced in the continuous culture, cells will be maintained in the exponential phase for longer duration for the large scale production of the metabolite. As a result of which, the exponential phase of the growth microorganism will be maintained for a very long time. In earlier culture, that is batch culture, it's a closed culture, only once nutrient will be provided. Whereas in case of the continuous culture, there is the further supply, intermittent supply of the nutrient takes place inside the fermenter through the inlet. At the same time, after the standardized time of growth of the microorganism, the spent will be removed, due to which the toxic substance will also be removed and the fresh media will be drawn inside the fermenter, and hence the cells will be maintained in a continuous growth or in the exponential phase for very long period. So this is continuous culture. Next, the another method is the fed batch culture. It is just the modification of the batch culture. In batch culture, usually only once nutrient will be provided. But in case of the fed batch culture, it's modified batch culture. Basically, it is the batch culture which is fed continuously. The nutrient will be added continuously to the same fermenter without the removal of the spent, without the removal of the original culture media from the fermenter. As a result of this, the continuous increase in the volume of the media takes place inside the fermenter. Cells will be grown for a little longer exponential phase in case of the fed batch culture compared to batch culture. But here the volume of the media will be increasing inside the fermenter as we supplement the media, fresh media inside the same fermenter. So you learned how the microorganism will be cultured. One is batch culture, continuous culture, another one is fed batch culture. So in this session, we learned the biotechnological importance of microorganism and the general basic design of the fermenter and how these microorganisms will be cultured for the useful metabolite for the betterment of human life. I hope you understood this session. Thank you.